All right, thank you, Pavel. That was great. My name is Rosemarie Asuda. I'm a scientific point of contact at Pacific Northwest Center for Cryo-EM, and I'm excited to introduce our next speaker, Paul Emsley, who we briefly met earlier. Paul is from the Medical Research Council Laboratory of Molecular Biology in Cambridge, and he'll be presenting on cryo-EM modeling with Coot and Moorhen. So go ahead and take it away, Paul.
Well, thank you. Thank you both for the invitation. Hopefully you can see, hopefully you can see, not my YouTube, you can see my presentation now. Is that right? You can see?
We can.
Yes, we can see the Coot.
You can see the kit. Great. Okay, I've lost my cursor. That's bad. Well, I'll see. Right. So I was asked what did I want to talk about, and I wasn't sure. So and the title that I gave was something like this, perhaps not exactly this though. But I will be talking about the refinement validation representation in Coot and Moorhen. What is Moorhen, you might be asked? More on that in a moment. So Coot is a desktop application for macromolecular model building, refinement, and validation. This is going to annoy me. I don't know how to turn it off. Okay. I'm seeing myself, and there's a big bar down the right-hand side of my image, my display, which hopefully you can't see. It is somewhat similar to, in principle, Chimera X, Pymol, VMD, and the others. The notable difference is that it was ugly, and the old version still is. And there was a reason for that. It's because CSP4MG was as a CSP4 project, and I didn't want to step on the toes of molecular graphics, so I concentrated on what I was more interested in, which is tools for model building refinement rather than making pretty pictures. Now, that's changed since about 2019. Well, 2008 is when the Python developers said that Python 2 was going away, and consequently that meant there was a change in the toolkit required for me as a consequence of that. So it's on the card since 2008, and it started in about 2018, 2019, COVID times. That's when it really happened. There's a big rewrite to make it work with the new graphics and new GUI, and separate the libraries so that they can be used with Python. More on that in a moment. So, this slide. It's over the years, 20 years now. Coot's 21, 22, something like that. There's a set of tools that are useful for working with macromolecules. Now, we are typically familiar with the graphics and GUI of Coot, and that has a refinement tool, real space refinement tool, and the addition of local distance restraints is something that will be discussed later, and difference maps too. But I got infected, by the way, a couple of weeks ago, and I'm not used to my voice may be still non-robust. I try not to cough though. So the program was split up into a front end and a back end, and the back end is called libcoot API. Not very exciting name, but there we go. It explains what it is. It's an API for access to the Coot libraries, and that can be compiled with WebAssembly, the tool from Google, and combined with a React GUI, and that's what Moorhen is. It also can combined with Python to provide Chappy, Coot's headless API. That's what Chappy stands for, Coot headless. And Chappy, in conjunction with Blender, has allowed us to develop BlendCoot, and more on that in a moment as well, towards the end of the presentation. So Moorhen is a Coot on the web. That's what it was initially designed to do. What that I needed to do is split up Coot for the calculations and representation. Now, this is exactly the same split that was needed to get Coot working with VR as well, or any kind of external representation. So that there's a maybe low-hanging fruit there for representation of Coot molecular properties in VR at some stage. Although that's less trendy than it used to be. So there's this project called CSP4 Cloud where one uploads one's data, perhaps even from the synchrotron, so no transfer from your local machine is necessary. The data are then put into the CSP4 Cloud, and that needed a tool where one could manipulate the models and do validation without downloading it. So it's all done on the server. And Moorhen provides that functionality. So as I said, we take the Coot libraries, which are now split off away from the GUI, and add into it a modern React-based interface in the web browser. It has more extensive functionality now, more than just Coot on the web. It is a React component, so you can get it from npm if you're interested in that. And we've been working on improving the way Coot represents the structures, and they may be useful for figures. And there's a gallery, so if you go to moorhen.org/gallery, you can see some examples. More on that in a moment. I've got a screenshot. So like Chimera X, the move from Chimera to Chimera X, Coot followed along the same path to some extent, at least in this first example here. Very much so. Instead of having multiple dialogues, which plagued olden Coot, more modern Coot now follows the one window approach, and so it removes dialogues or makes them transients if they couldn't get rid of them. But generally I tried to, as the GUI was rewritten for the new version of the widgets. So I'm using revealers, planes, overlays, switches, popovers, which is a modern way of using an interface. Coot's graphics by default are black background, and that works well in dark mode. It should be with some little effort, but not no effort. The icons need to be modified to work with light mode, but light mode should be a possibility too. Now, the way one uses the program is different, and it takes maybe a few minutes to get used to it. So whereas before you picked a tool and then picked an atom, Coot now is select first. So you put the thing that you want to operate on, very typically a residue, a chain, an atom sometimes, at the center of the screen, and then you choose the tool. And this is consistent with the way key bindings work. It used to be the case that you rotated the view and picked atom with left mouse, and now right mouse takes over the view rotation functionality, which leaves left mouse for clicking and picking, and you don't get these two mixed up. Well, that's the plan. Many people, well, I say several influential people who didn't like this. They wanted it to work the old way, so the old way is a possibility as well in the preferences. So you can set it back to both left mouse for view rotation and picking. I don't approve, but it's there. 22 years old, many tools added, and the interface is busy, so it covers validation, B factors, Ramanujan's, density fit analysis, and you can see all of the other tools there. But the tool that I have spent most of my time on, perhaps the majority of my time, is the real space refinement. It is perhaps the major feature of Coot, and has taken a lot of effort to bring it in to functionality, because there's a lot of background that was needed to handle dictionaries and things, and can create restraints quickly. So that took some effort. We're using standard CIF-based libraries, so we can use the monomer libraries from the dictionaries from the CSP4 monomer library, or global phasing, grade 2, and Phoenix Elbow. So they're all interchangeable because they use the same similar CIF-based file format. The idea then is we use the molecular descriptions there to create target functions which we can combine together and create something that can get minimized in a minimizer. I mentioned it earlier. The one in Coot is a first order minimizer, not as sophisticated the one in RefMac, and including links and modifications now, or more recently. And you can do things like dynamic and interactive refinement. Maybe we'll have a time to look at that. I've given tutorials or seminars in the past on Zoom, and when I tried to do a demonstration, it didn't work very well. So most of this will be just normal standard PowerPoint-like. But maybe we'll have time to have a quick demo at the end. I don't know if it'll work because the frames per second make it, or have in the past made it problematic. But Pavel's animations look very nice on my screen, so maybe it'll work. We'll see. So we know from the dictionary bond length and angles and chiral centers and which atoms are in a plane, because we have some understanding of aromaticity and delocalization. And we can find analytical gradients from the mathematical description of those geometries and combine those with the gradients from the map and use those for minimization. Now, I said we know the chemistry, but what do I mean by that more precisely? How is this chemistry information created? And what we use in our community, in our lab, is ASTRUG. Having said that, by the way, Pavel, I just thought I wanted to comment on your slide about the number of people using Phoenix Refine to do their refinement. I think the majority of people in our lab using Phoenix Refine, just by the way, which I think you may find amusing. Right. We use ASTRUG to generate restraints, and ASTRUG uses COD, the crystallography online database, as a repository, a database of crystal structures. A useful outcome of that is that we don't have to worry about entanglements with the CSD license. They don't own the intellectual property of our structures. That's good, or yours, more importantly. And creating restraints, complicated process. It's been a program that's been in development for just as long as Coot has, really. And initially used to use only the organic set, so carbon, nitrogen, hydrogen, sulfur, boron, but not metals. But recently this has been added to the CSP4 suite in a program called Metal Chord if you want to sort out your metal geometry as well. Right, and you got the references there. So how does the atom typing in ASTRUG work? Basically, it's second order neighbors. So you can't see my cursor. This is not going to be very useful. Okay, so basically H1b, it's a hydrogen attached to a carbon, and that carbon is attached to two other hydrogens and an oxygen. So that gives you second order information, and that is used to describe the local chemistry of the atom. And there are more complicated atom descriptions or atom types. So, for example, the C9 is in that ring system in the top left-hand corner here, which is a combination of 5 membered rings and 6 membered rings, and the atom type looks like this. Once an atom type has been decided upon, then in the small molecule crystal structures, which contains around 1 million now, not quite 1 million, but that sort of hundreds of thousands of crystal structures, that they can be analyzed to calculate, in particular, the bond lengths and the bond angles. And they can be used to inform our refinement. Tristan may talk about this as well, perhaps. It was briefly an example a few years ago now that was problematic. The cispeptides and Coot perhaps was a contributor. I won't dwell on this too much. The idea was that if you wanted to, you could drag around the atoms in real space refinement and push the nitrogen around so that it was on the same side of a peptide bond, well, between two C alphas as the CO. And if you then applied planar restraints on that, then you'd squish it into the cis conformation. Coot warned you, and it was my view that if you didn't know what a cis peptide was, you had no business pressing the OK button. But apparently some people did, and there was an issue of cis peptides. Now, Coot, as it's preparing its restraints, it analyzes the peptide bonds, and if you have a trans bond, most of them are, of course, then it will add in an additional restraint on omega to keep it around 180 degrees. It very much objects to being around 0 degrees and will try to twist itself so that it's 180 now. This is what it looks like in Coot with the white background. I can't really see. It's obscuring my slide here. So red is for peptides that are cis peptides that are in front of residues that are not proline. Green is for cis peptides that are in front of a proline, and twisted trans is yellow. So sometimes you'll see red, and that's often interesting. And sometimes you'll see green, and that's often the case that proline has been added by evolution, because it's energetically not disfavored as another residue type would be. Right. So, but when you see yellow, well, I've never actually seen twisted trans being in a real structure. So it's always the case, at least in my experience, which is, well, I have some experience of looking at protein structures. If you see a yellow ribbon, then that means something needs to be changed. Of course, we live in an AlphaFold world now, a post-AlphaFold world. It used to be the case when one was doing model building in Cryo-EM maps, then the actual working out where the atoms go was a considerable task. So that's so-called de novo model building. It's no longer so important as it used to be. Now what we can do is find a model that may or may not be close, and then plonk, plonk, and then do a rotation translation search to fit it into the local area. It's very frequently the case that you know whereabouts in the structure you want to add in this extra domain. Phaser has this tool called EM placement. You may hear about it more. There's an interface to Chimera X. I'm working on an interface in Coot. I have some problems with installing it though. I'm waiting for other people to do an update. Right. So if you look at the Coot CryoEM tutorial, part of it is to fit an AlphaFold model. So we look up the UniProt ID and then download the model from that database, the AlphaFold website, and then you follow the tutorial and you can add it into the map and then add that model into the rest of the model. There's another part of it which is typically what one would do is you extract a domain from a homolog and then fit that to the map. And then there's another part of the tutorial, part 3 of the tutorial, is building a hormone de novo. In this case, it's GPCR receptor, and the hormone is alpha helical. And then part of the part of the modeling operation is to assign the sequence. Every part of this is followed up with a real space refinement. Now, we have talked about how we can generate distances and angles and chiral centers and so on. So that's standard geometry that can be used, where we use dictionaries to inform our understanding. We're also in CryoEM, I find this is very useful, is local distance restraints. So this was first introduced to me by Rob Nichols and team. And that's his paper and his thesis. So the idea in his thesis was crystal structures where we had you have a high resolution form where you have the ligand bound form, and then this weak structure is a low resolution map that doesn't have the ligand, but you want to use the information from the high resolution structure to inform the low resolution refinement, and we do that with local distance restraints. In this case, we can do structural similarity comparison. That's where the idea is, but very typically in Coot and CryoEM we do self restraints. So we use starting model restraints. If we're looking at a homolog, or the same structure with and without a ligand, what we can do is show the distances from the reference and compare it to the distance in our actual structure, and then use that to color up the distances between the atoms. And what we can see here is that when there's no ligand bound, there's a set of differences. That's what the purple lines are. They're longer in today's model, which doesn't have the ligand, and so the ligand binding site's opened up a bit, and the distances between these atom pairs are longer, and so they can be edited, removed if you like. But the weighting system of the scheme, as you'll see in a moment, is robust to allow this kind of long range distance difference and still be robust. So comparing least squares and what's called Gebel-McClure, that's what we're using, a Gebel-McClure modifier to the target function. So the idea is that as we move away, I can't move my cursor, which is annoying. As we move away from the target distance, the shape of the function changes. So the further we move away, then the more the target function doesn't care, because the variation of that value on the y-axis is small. Right, so if we move a little step, so if we're oh dear, I wish I had my cursor. We're moving from here to along a bit, so further away, and when we have a least squares target, that's a big contribution to the penalty function when we're using the Gebel-McClure modifier. M estimator, as it's called, then it's adding a small amount. So the further and further we move away from the true structure, the less and less we make a contribution to the target function. It's not zero, but it's increasingly less. And what that allows us to do is to keep the local distances consistent. Lots of local distances restrained, but allow a few to be kind of broken, because after some time they just give up contributing to the target function. So I think that says large contribution and small contribution. I can't see because it's obscured. Right, so let's have a look at an example of this. I'm going to right, I'll try to end up at 35 minutes past. Let's see if I manage it. So this is an example from my colleagues from a few years ago, in pre-COVID times. Cleavage and polyadenylation fracture CPF. So at the time, it was a nice high-resolution reconstruction, and how would one go about using a homolog? At the time, it was relatively oh dear. there was not much other structures known, but later on there was one that was published a few months later. So this is what I've done. I've taken their model and plonked, right, just added it into this space. And what you may be able to see is that it's completely the wrong way around, but the centers more or less match. And using Coot's rotation translation tool, which is multi-threaded, so that's one of the innovations that's happened since COVID times. I learned about CUDA and multi-threading. So now we can use that multi-chord processes to do lots of tests of hypotheses of rotations and translations. And I changed the colors here, so hopefully we can see the way these things fit better. So after it, after what's called jiggle fit, it finds a solution. Now, the model that we start with doesn't exactly match our experimental density. If it did, there'd be no point in doing the experiment. So there is some differences, and what I'm going to show you is that we can. So I'm just turning the model around a bit. We can use Coot's real space refinement with local distance restraints. Now I call this morphing. Now Pavel may disagree with that terminology. We should have a discussion one day. I call this real space refinement morphing. So there's a local distance restraints, and what we can do is track how they're changing in distance by coloring the model as we fit the model. So this is a few screenshots. As I said, the refinement, it doesn't work well, or the animation doesn't work well. So I've got a few screenshots. So this is me taking a few samples of screenshot as the refinement progresses, and you can see it's fitting in there. And in reality, this is taking a minute or two, about maybe 80 or 90 seconds, if I remember. And then you can see it's really quite, it's done its job. It's quite well fitted. If I change the contour level now, you can see that each of the strands is well, as far as we can tell from this representation, is reasonably well fitted. So this is an example from Maya Toff and team, and I think Greg Pintilli also used it in his recent paper, a Q-score, I think. So the idea is to take a GrowEL domain and try to fit it in, and it doesn't fit in. You can see those helices are missing on the left. So after some real space refinement with these local distance restraints, it takes a few seconds to refine it. This is a Coot screenshot, by the way. So as well as we're doing long range order, getting it right, because we're allowing it to be corrected by the experimental data, we can look at the details of the interactions, so side chains. What do we do about this? For example, here we have a modeled, is that a glutamic acid? Aspartic acid. And then there's a red blob. Where's the red blob coming from? The red blob is coming from Servalcat. Oops, where has that slide gone? Oh, I've missed a slide somehow. So now there is a non-blocking interaction with Servalcat. Non-blocking means that the user interface doesn't stop while the refinement is running. And what that means is that it can take the current model and compare it against the reconstruction and give you a difference map kind of live. It's not within a fraction of a second as it would be for crystallography, but it's within 20 seconds, which is better than nothing, or better than it used to be without this useful feature. And what that means is you can do B-factor modifications on sort of on the fly if you can tolerate 20 seconds per update. But certainly when we look at other side chains, we can wonder or worry about radiation damage, as it might be in this case. in this case. So what would one do here? Leave the sidechain on and delete it? My personal opinion would be to keep the sidechain there and let the B factors go to many hundreds. Here's an example that isn't radiation damage. It is, this is a structure from the PDB. I didn't put the code on, but I could find it if needed. So yeah, so they've done some molecular dynamics, but they didn't allow enough flexibility of this tryptophan here to find the correct orientation. Had they had this difference map access FC sort of map for CryoEM, then they could have straightforwardly identified a rotamer flip for this, a rotamer flip that the molecular dynamics was not able to perform. It couldn't escape the local minimum. Maybe even we could look at multiple conformations in Cryo-EM maps these days, being informed by this difference map. So we can use refinement in Coot for and model building, and one of the other things that it does is validation. We've heard the validation is boring. Maybe it is. Maybe it is. Maybe not so much for me. So there are some validation tools that you may be familiar with, non-body contacts, or clashes, as they're called in the mole probity world. Geometry analysis, how we can say how well does a ligand, for example, compare to its restraints. And there's an implementation of the Q-score now in Coot and Moorhen. And so we're more than 99% correlated with the Q scores that I tested with Chimera X. And updated Ramachandran plots as well. That was thanks to Andrea Thorne and her team, and Bernard Locamp, who put them into Clipper and made the GUI in Coot. Right, I'm going to skip over rotamers just to let you know, though, that CryoEM maps have influenced the algorithms that are in Coot. So I won't spend too long on this. It used to split the residue along the main chain. It turned out this was a mistake. If we use the formalism of the Richardson's team, called the back rub, which is used to be also the name for Google before it was Google. We can then use this mechanism to find high probability rotamers in low resolution maps. So this is scattered across the back rub rotamer, and then we can compare and contrast. So this is the old method. We collapsed the main chain, and the new method is using pink here, and the solution here is just to, you can see that the main chain is hardly moving and the side chain is twisting a bit, slightly out of density. So we normally were happy with, we are familiar, at least as people who look at CryoEM maps, we're familiar with the idea that side chains of proteins that we would expect in high resolution and crystal structure maps, where would you expect sidechain atoms to be in density in Cryo-EM maps? We're very familiar with the idea that they frequently are not. And so this kind of solution here, where we have it a little bit out of density, is not so bad. Anyway, we will then feed it to the real space refinement tools, and either whichever one you want, and that will tidy up that slight misorientation there from the rotamer fitting. And this is called back rub rotamers in Coot. A recent addition to Coot is Coot and Moorhen and Chappie is ligands. So I've implemented the Clark and Labute, well, at least some of it. A good part of it, not all of it. Ligand series is missing, where we can look at the protein ligand interactions, fingerprints perhaps, and make, one hopes, attractive looking figures. There's a few examples here. And now I will spend a few seconds showing you some I call them pretty, pretty pictures. Right. So as I said, my interest is mostly in the scientific developments of Coot, and graphics was not a priority. It has been more important to me recently, partly because it was forced on me, and I thought, well, seeing as I'm in the area, I might as well try to learn it. And the way one learns graphics, or OpenGL, is to write a game. All the tutorials are written for game authors. There's no reason to write to learn OpenGL unless you're writing a game, right? So I went through the tutorials and learned how to write, provide the infrastructure for writing a game. I didn't know that there were some things that I didn't need to know. And several of these things have their own shaders. They're programmable, and you can preferentially use your own shader instead of the shaders that I have provided. So there's this thing called Sponsor. There's a model that you can download, and it tests rendering engines. So what we're looking at here is a thing called ambient occlusion, and it's a big thing in games because it makes the representation of geometry more realistic. So the only thing that we're looking at here, we're not looking at shadows or colors or texture mapping or anything like that. Only the ambient occlusion, and that gives you a good well, I put it to you, ladies and gentlemen, that you get a good representation of where things are in space using the ambient occlusion engine that's now in Coot. So I was basing it off these models. I was basing my engine off of the models that I downloaded from the internet, these boxes. I wrote by hand, but the models there I downloaded. Because they're simple, because they consist of only a few hundred vertices, it's very easy to start up the program and test it rather than looking at a whole protein. But Chimera X allows one to generate surfaces and export them as GLTF, and that's what I did. This is a Chimera X surface here, exported it, and then loaded it into my engine here, just to make sure that it did sensible things with proteins as well as D&D figures. Right. Models and maps are now 3D objects that interact with the light in a way that they didn't used to do before. It was very simple. It was surprising how useful it was, but it's better now. It uses instancing, which is the way game graphics cards developers or manufacturers want you to interact with or provide geometry for their cards, because they're very fast at handling instancing. And there's a thing at the bottom here, credit to Tristan, who first showed this to me and others, updating maps and how useful they were. And I implemented that with the help of Kevin, as he was at the time, Catherine now, Cowton, and yeah, that was very helpful. So this is what it looks like now. Dark mode, we have ability to have ribbons there. You can see, you can perhaps also see that the map is interacting with the light, and so it's a better smooth. You can see the shape of the density better than you could previously. The Ramachandran plot is interactive, and the geometry is interactive as well. If you turn on the analysis, you can see Ramachandran probabilities, Rotamer probabilities. There's I call it a head-up display, and health bars in the top left-hand corner. It's not actually health bars, it's damage bars. So the idea is there to allow you access to move around the structure looking for problematic geometry. Like you've been shot in the arm, and you need to get your med kit, and so that's the idea. So you can navigate to that place and then fix the problem. Right. Right. So as part of learning about OpenGL, one learns about frame buffers, and that's how screen space ambient occlusion has been implemented. And there's other frame buffer techniques that allow one to do outlines and blurring and shadows, and these have all been added into Coot now. So here's an AlphaFold model, PL colored by PLDDT. And this is ambient occlusion. I colored the ligand there purple and made the hydrogens blue just to show you that, well, I was playing around with the color schemes and the SSAO at the same time. We can do things with changing the what's it called, the shaders. We can get different lighting effects like Goodsall effect, Goodsall kind of mode here. Ambient occlusion and B factors, and we have hopefully you can see that this is sharp at the front and then blurry at the back, maybe. So this is a structure that I got from the Chimera X paper. Yeah, and so we can do Gaussian surfaces. I just implemented their method. And it looks okay, I hope. And it rotates perfectly fine, at least to me. Carbohydrates and spike protein, which you may be familiar with after all these years of seeing it. Other virus structures. We can color by chain, and we can map colors from we can have two maps, right? One for surfacing and the other one to provide some value, correlation coefficient, or occupy or resolution. One might use those kind of things for representing this kind of structure. So also a perspective and blurring and Fresnel lighting. So you can see the density contours, it's a black background, the density contours are black, and there's blurring going on, but there's also Fresnel lighting going on as well. And this is very interpretable, even though it's black on black. Well, I put it to you, ladies and gentlemen, that you can interpret the ligand density here. It's my view. Now you use it yourself and determine whether you agree with me. So this is again COVID times from my colleagues who made this high resolution reconstruction in CryoEM. This is something published this week. Andrew Ward and team, where they had these antibodies but were not well modeled. They didn't put a model in the constant domain, but with the easy access to sharpening and blurring now, we could create a reconstruction or a variation of the reconstruction that allows us to see where the constant domain is, and you could have modeled that if you'd have wished. It looks okay in a white background, I would say. Of course, you decide. Anisotropy, that's recently corrected. So the people at Global Phasing saw that I'd been making a mistake with the representation of the thermal ellipsoid. So that's all fixed now. And you can do small molecule crystal structures and ORTEP-like representations. When you download structures, you also get this PAE plot. What Dorothy would like is to be able to interact with this plot and then carve out domains. I would like that too now, but I haven't managed to do it yet. So hopefully by the time we meet in Cold Spring Harbor, I will have done it. Right, so last thing is to use Coot for interaction with Blender. As I said, the graphics have been stripped out and it allows us to add in extra heads. One of those heads is using the GUI of Blender, and so the tools of access, you have access to Coot tools in the Blender interface. So you can download structures and create ribbon diagrams and ligand representations and do masking and Fourier transforms and filtering. Yeah, so if you don't know about Blender, it's increasingly popular and being used commercially. even. So Lucrezia has taken libcoot API and added an interface between Blender and Coot. It looks something like this, where you can download structures, or just upload your own, of course. But there is I'm not sure competitive is the right word, but there's another project called Molecular Nodes, Brady Johnson, that plays in the same area. You might like to look at that. That's more easy to access. I suspect it's in their what's they call it, the shop front or something, that's easily available from Blender. So here's a few examples. Tin barrel, and then that was that structure we were talking about here. So spooky seafood, I think I call that. Carbohydrate, and where we have a transparent density, and it's difficult to represent carbohydrate. And as I said, we've got masking as well, so we can mask out the density for the ligand and represent that differently, should we wish. And you can do atom selection and turn those. We've got the the hemes here. You can't really see. I'm sorry, my mouse is missing, but you can look carefully. You can see some hemes, and they're lit in white rather than ribbon diagrams. Again, we can do RNA and DNA and reduced representation. Again, I was inspired by Chimera X to get this base representation. So those are the things that we are available in Blender and Coot. There's some screenshots available in Moorhen, and the graphics in that are still in development. Not that, and that's actually not been written by me, but here's a GPCR receptor, and this is how it looks in your web browser. Perfectly rotatable, you can see a whole reconstruction. There are some programs that have particularly bright colors of green, which you can reproduce in Moorhen, should you like that pie mole-like figure. Again, masking and van der Waals radii. Sorry, not molecular surfaces available. Electrostatic surface is not available today, I think. Again, anisotropy is, because it's written in the library, is available in Moorhen as well as on the desktop. And we have ligand interactions, so van der Waals surfaces. You can see some colors where they're getting close to the protein, and if it's a hydrogen bond, it will be represented in purple. And we can see the chemical features of a ligand, so hydrogen bond donors, aromaticity, and turned on the distances with the interactions with the protein as well. And this is, one hopes, helpful for ligand design. Ligand binding analysis, ligand design. I mentioned that there was a gallery, and this is how one gets to it. This is what it looks like. How you get to it is you get to moorhen.org/gallery, and you click on an image, and it loads up that representation for you, and it sets up the GUI so that you can see what the icon shows you. So how do you get it? The place on the web that it is the way I do it is Google LMB Coot, and it finds it. You can also get it from Flatpak and Homebrew, CSP4, fingers crossed, Phoenix not too soon. No, that's not right. Soon, not too long. And Moorhen, of course, is a website. You just go to the website. And if you want to see some videos are difficult to make, and I'm not very good at making them, or kind of slow at making them. They do take some planning and editing, but there are a few, and if you want to see them, they're on YouTube. Right. Thank you to my collaborators in this, the people at the lab. Useful discussions. Global Phasing has paid for Jakub Smolski, who's been working on the ligand interface. And the Moorhen team has been fun to interact with, which we do completely virtually. I don't think we've ever met in person. Yeah, so that's funded by CSP4. Stuart McNicholas of CSP4 MG fame. Philomeno has now moved on to more lucrative things. Martin Noble has provided the surface and ribbon diagrams, and Lucrezia is now part of the team in at the LMB. Right. I wonder if we've got a couple of seconds to have a look at the old Cootie McCoot face in on the desktop. So let me just run it. And let's see. I don't know whether this is going to work or not, but I will fire it up. That was a mistake. Can I? Oh, I can do it now. Oh, I should have done that an hour ago. Okay, so let's go and load the tutorial, and we can see this is loaded the difference map, and there's an issue here. Right, so if we turn on what should we do? What should we do? We could just turn on real space refinement. So there's sets of operators down here, so it's different, right? So if we do RSR sphere, real space refinement sphere, we now have some textures that unhappy, right? The idea is that what we're looking at here is unhappy interactions between your protein, and as I pull on the atoms, then they disappear. I kind of push it in, they get unhappy, and I pull it away, and then they, like little balloons, they disappear and pop. If you turn the sound on, they go pop, 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 pop. It's quite fun. Maybe not for your colleagues, but I don't know. I find it fun. There is a way of turning the sounds off. You're not forced to listen to the sounds. So this Ramachandran plot is interactive. So if I click here, then we can see some problem. So if I'm just to accept that and then turn on the refinement, we can see red balls for bad Ramachandran and green dodecahedron for good rotamers. So I need to interact with this, and I can yank around the atoms again like this. I can create all sorts of problems, and you saw the health bars moving around here. And what I need to do is to flip that oxygen down to over here somewhere. So I just do pep flip this. It flips it down. You can see it change color. And this is a yellow here. You see, this is a yellow rotamer. So if I bring it to the center of the screen, and then I do background rotamers, it fits in and changes the color. Right. So I'll end there, and I will take questions.
Thank you very much, Paul. That demo at the end is so fun with the angry faces coming up and popping away. That's extremely fun. All right, I'll take a few of the questions from the chat. So first of all, in general, now that we've heard about both Coot and Phoenix, I'm wondering if you could talk a bit about how one can use both as parts of a larger workflow.
Yeah, that's a question that we are enthusiastic, both ends, to pursue. So we have the infrastructure to. So the answer to your question is I don't know today. We will do a better job working together to have a smooth interaction in future. So how best to do that, I don't know. Don't know today. But we do have access to two-way information. And merely pressing the button on the Phoenix interface and loading up the structure, and then going to if you have Phoenix or Molprobity validation, and you can click it in the Phoenix GUI and use that to navigate to a particular place in the Coot structure. That's all very straightforward, and we should do it when we get together. We should just do it when everybody got that. The current problem is making sure people have the program, and once we do have access to the program, I have Phoenix, they have Coot or modern Coot. We should sort that out. So the answer to your question today, it's not very good, but it will be lovely in a few months.
Okay, great. That's exciting to hear. There's a question about how Coot can be used to validate placement of metal ions.
Right. There is this thing called WASP, and the authors are now escaping me. Can I Google it in time? There was a metal protein analysis. No, 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 no. Yeah, I've forgotten his name. Sorry. I've forgotten your name. So Coot implemented that method. I read their paper, implemented their method. I added symmetry so that we can have symmetry-related atoms, which I don't think was in their protocol, but it's added in Coot. And you can do, it's called electron shielding analysis, and you can use that to determine what is a water and what is a metal. Better done, I think, these days, at least in the, have a look at the metal cord paper and see whether that's helpful. It certainly produces restraints, and that is a different question to the one which you asked about, which is about validation. So I that's as much as I can give you at the moment. There's a tool, and you can use it. It's old. It's based on a tool that I can't remember. It's called Wasp. I don't remember the author. Yeah, that's it.
I think it's Enrico de Serra.
Yes, thank you. Thank you.
All right, I'll ask one more going into our break until 10:45, but then continue covering questions during that time if you want to stick around. But there's a question about how to make a difference map in general, and then if one needs to worry about scaling one map to the other, how that can be done.
Yes. So what one does is the problem with scaling is worthy of concern or consideration. What we could do is to set RMSD so that it's about the same. So 2.1 in one map is 6.4 in another, and you set the levels to that they look about the same. It's within you can do it using the scroll wheel within a percent or two, and that works fine to work, at least in my hands, to allow you to find the you divide one RMSD by another, and that gives you a scale factor. A more modern way is to do power scaling so that you have, and that allows a variation in or possible difference in B factor. The one map gets scaled to the other over the resolution ranges, and then you can use that to calculate a difference map. Yeah, so that's a more modern way of what I would do is power scale it first and then calculate the difference map. Add two map. I think you might need to add two maps together with -1.
Great, thank you, Paul. Yeah, we will continue to address some of the questions in the chat, but there is a break now until 10:45. So if you need to go stretch your legs or grab a coffee, whatever, until the next talk, you all are welcome to do that. And Paul, I will add there's a comment that just says, thank you for making modeling fun, which I concur. That's certainly apparent in Coot. So thank you.
There is a question about whether binaries are available for Linux-based OSs.
Yeah, Flatpak is the best way of doing it, I think. But I've been in touch with some people who are Debian fans or Ubuntu fans, and it's been part of the operating system as well. So that's apt-get install coot should just do it. Are there good RNA modeling tools in Coot? Well, okay, so there is. You can make what used to be called Ideal, but I realized after some time, later than I should have done, that it wasn't as the models of RNA that Coot was producing wasn't as good as I had imagined that they were. But you can make some RNA models, and then what you do is point it at the density, maybe even YouTube. I could show you. I could show you Coot RNA. There's a YouTube video. Yeah, this is the one. Coot with RNA. So it's narrated by
Here is noted the original map.
Anna.
4225.
Anna, right, Casaniel. So the idea is you turn on local distance restraints, and then you can see it's a long way off to start off with, but the map is just good enough, and the local restraints just make sense to fit, and it fits the model to the map. This is a few years ago now, so the computers are going a lot faster than this refinement is. There we go. It's just doing its minimization, and there we go. And it's refined in. So local distance restraints, in addition to base pairing and base stacking, do a good job, I think. That is, if you're talking about RNA modeling in the light of CryoEM, like this is a CryoEM map. There you go. So it fits reasonably well. And then if we can compare it to the reference structure, you can see it's, I didn't get the sequence right, but you can see that it's done a pretty good job at getting the thing.
Well, we can't
Oh,
see the YouTube window.
you can't see it because I'm not sharing. Oh no! Oh, that was, thank you. Sorry, I'm wasting your time.
Oh, no. At least we know where to look.
Let me okay, let me just, if I've got 10 seconds. Where's my zoom? Zoomy zoom zoom. Is this what I want to do? Share? Is it this one? Right. If I take it back
Yes.
to about here. Right. So local distance restraints plus base pairing plus stacking. And we start off with something that, yeah, that's about right. Right, all atom refine. The GUIs are all completely rewritten now. Yeah, so it's moving slowly, so it's moving through. It'll be 3 or 4 times faster these days with the modern computers. And this is all interactive as well. So if you wanted to yank it, to use the technical term, if you wanted to refinement, then yeah, you can do that. There you go. So now it's fitted. And then if we compare it, you can see. Right. Okay. That's what I wanted to show you.
So kind of a general question, when do you advocate for building or refining models into difference maps as opposed to non-difference maps?
I this is not a question I've heard of in the context of CryoEM. It is a question that some people have asked for crystallography. I've never do it. It seems to me that the difference map is more noisy, and I'm quite happy to just refine into the 2FOFC in crystallography or the Cryo-EM reconstruction. Very frequently, I find that they need they're over sharpened to my crystallography eye, and they need to be smoothened and resampled just to get it to look like what a macromolecule should look like. Certainly, and it's also more aesthetically pleasing when it's sharpened. Sharpened and resampled.
Along the lines,
I don't do it. You can try it if you like.
along the lines of the Linux question, what is the best way to install Coot on a Mac?
Homebrew.
Okay.
If it's in Homebrew, then it's a brew install Brewski bio Coot. Brewski / Coot.
Are there any tools in Coot for working with lipids?
Not today. Not today. It's something that needs to be addressed and is a proper research project. I'll see if I can get it funded.
All right, and one last one before we wrap up the break. One person is commenting that after manually adjusting the positions of atoms in a ligand and then subjecting that to real space refinement, the atoms moved out of the density. What would be your advice?
I don't. The devil's in the detail. I would have to see it. It might be that the chirality of the model is inconsistent with the chirality of the restraints. Maybe on an atom or two. That's something that I would look at first if I saw it behaving oddly. Maybe non-body contacts also.
If one wanted to just take a shortcut and constrain those atoms, is that possible?
Yes, you can anchor them. There's an anchor tool that's under. If you look at real space refinement menu item, you can set an anchor, and that means that atom is not allowed to move during refinement.
All right, well, thank you again. And I am going to turn the floor over to Steve.
All right, thanks, Omar. Thanks, Paul.